And I told you with Agrobacterium tumefaciens, it's possible to transform more than 60% of the plant species. When we are not able to use Agrobacterium tumefaciens, it may be possible to use some other agrobacteria, mainly Agrobacterium rhizogenes. At the end, there are still few plant species that you will not be able to transform using agrobacteria. So in this case, how can you do? Are there alternatives to make GMO plants? Sorry, yes. So we may use other approach to transform the plant that cannot be transformed with agrobacteria. And those approach are the following. You may use protoplast. So I told you it's possible to make protoplast out of some plant tissue. So again, it's not possible for any kind of plant, but from most of plant species, you can make protoplast, which are plant cells without a cell wall. So to make protoplast, what we do is to digest the plant cell wall using cell wall degrading enzymes. And it will be possible to insert DNA into those plant cells by different approach. You may use electroporation. So as we do for example, Escherichia coli, we can transform plant protoplast using electroporation. We will use an electric pulse to insert DNA inside the plant cell. You may also micro-inject, that's what you can see in this picture, DNA into protoplast. And finally, you can use detergent, which will kind of destabilize the plasma membrane to make DNA enter into plant cells. So this is an alternative to the use of Agrobacterium tumefaciens. Still, it's not so easy to make protoplast out of plant tissue, and protoplasts are very fragile. So it's an alternative which is used only for some plant species, I would say. And then, and I told you previously when I spoke about in vitro culture, it's not so easy to regenerate a whole plant from protoplast. So this alternative will only be used for some very, very specific crops that you may not be able to transform otherwise. Another alternative to the use of Agrobacterium tumefaciens is particle bombardment. The idea is to use what we call a gun, which is in fact a compressed gas gun to bombard plant tissue with some particles which are coated with DNA. Okay, it's microbeads that will be coated with DNA that you will introduce into plant tissue. So it's what we call the biolistic method. It used bombardment of particles to insert tDNA into plant cells. So this has been used a lot to transform crops that may not be transformed with Agrobacterium tumefaciens. but really, I would say a lot of progress has been made in the field of in vitro culture that allows to use Agrobacterium tumefaciens to transform more and more plant species. So the use of protoplasts, the use of biolistic, it's really specific to only some plant species. But it's methods that have been used, and it's interesting to know them. Okay? Then, the protocol I presented you, I go back. This protocol is meant to regenerate a whole transgenic plant from some small transgenic cells, I would say, some few transgenic cells. So it's a protocol which is meant to generate stable transgenic lines. The same with the flower dipping method. The idea is to generate stable transgenic plants. But you may use also Agrobacterium tumefaciens or Agrobacterium rhizogenes only to transiently express a transgene into some plant cell. Okay? And that's, for example, the method that are presented here. You may want, so then it's not meant to produce, I would say, transgenic crops. It's more a tool that will be used in laboratories to make functional genomic. You may use Agrobacterium tumefaciens to transiently express a plant gene into some plant cells. And there are two methods I want to speak about. It's the agro infiltration, and the second method is using protoplast. When you do agro infiltration, what you will do is to infiltrate. That's what you can see here. Agrobacteria. So this, what you see in the syringe, is a suspension of bacteria, a suspension of agrobacteria, and you will infiltrate those agrobacteria into the leaf. And you will not, from those infiltrate cells, you will not will to regenerate a whole plant. The idea is that it's only to transiently express here in the epidermal leaves of a tobacco plant, you will only express transiently a plant gene or a chimeric construct, allowing, for example, to localize a plant protein into those tobacco cells. So it's a tool meant to do functional analysis and mainly to do subcellular localization experiments, for example. That's what you see here. And you can do the same with protoplasts. You can transform them doing electroporation and not will to regenerate using in vitro culture a whole plant out of those protoplasts, but you may only will to observe those protoplasts that are transiently expressing a transgene. Again, to do some functional genomic. Here, the idea is not to produce a transgenic plant. The idea is to produce a transgenic cell that you will be able to observe in vivo. So it's a tool to do functional genomic. So it's a transient expression versus production of a whole transgenic plant. Okay? The take-home message, when you want to generate a GMO plant, you will use mostly, but not always, Agrobacterium tumefaciens. Okay, but you may use some other approach, such as particle bombardment. The idea is to insert into plant cells a tDNA. And this tDNA you will have produced using molecular biology method to make this tDNA carry a gene of interest and a selectable marker. That can be any kind of gene of interest, and we were speaking about a few selection genes. Once you will have inserted this tDNA into one plant cell, you will need to select this transgenic cell. And you will need to use in vitro culture to regenerate from this plant cell a transgenic plant. You transformed a plant with one specific goal. You had one goal, I don't know, it was to, for example, to test the ability of one isolated gene to make plants, I don't know, resistant to a stress. So you will need to evaluate if this transgenic plant is indeed able to resist to a stress when it carries this gene. If this is true, you may apply this observation to a crop, a variety which is a commercial variety. Okay, so that's a kind of protocol you will use to use genetic engineering to improve a plant. Okay, that's what we spoke about in the last hours. Now I want to move to the next step, and I want to introduce you to the most emblematic GMO plants. Transgenesis has already been used to improve plants, and I want to introduce you with the most famous example of GMO plants that are now commercialized. Generally, genetic engineering has been used for agricultural purpose to improve plant resistance to disease and pathogen. Okay, most of the commercialized transgenic plants are meant to be resistant to disease and to pathogen. And I would say that the second most used purpose of GMO plant, it's resistance to herbicides. Okay, so the huge majority of commercialized GMO plants are plants that are resistant to disease or resistant to herbicides. And of course, other transgenic plants have been produced that are more resistant to abiotic stress. For example, more resistant to salt stress, more resistant to drought, more resistant to frost. This has also been used to improve plants. Few plants have been genetically modified plants have been generated with improved nutritional values. And I think the best example, it's the golden rice. And this we will have the opportunity to speak about when we will do the tutorial. And then also plants have been created with some modified taste, modified aroma. Also plants transgenic plants have been produced that are more able to resist to post-harvest processes. And finally, it's the case for cotton, for example. Plants have been improved for the quality of their fibers. So those are different examples of the use of genetic engineering to improve crops. The third generation of transgenic plants that were presented on the market were plants that were able to resist pests, to resist biotic stress, and plants that were able to resist herbicides. One of the first GMO plants that was commercialized was a plant able to resist to insects, and I will be speaking about it later. Also, corn has been so cotton and corn resistant to insects have been commercialized, and those transgenic plants resistant to insects are carrying what we call the Bt gene, a gene allowing production of a toxin for insects, which is a toxin normally produced by a bacteria, which is Bacillus thuringiensis. I will be speaking about this later on. The second type of first generation GMO plants were the plants allowing resistance to herbicide, and this herbicide was Basta. And this has been transferred to a lot of crops: soybean, corn, wheat, sugar beet, and so on, have been generated that are resistant to herbicides. I want to say a bit more about the use of the Bt gene, which has made it possible to render cotton, corn resistant to insects. This Bt gene is normally, it's a gene that was isolated from a bacteria, which is Bacillus thuringiensis. And this bacteria is expressing the Bt gene, and it allows this Bt gene to produce a toxin. And the idea was to introduce this Bt gene into plant cell to kill some insects. And in fact, if you spray this Bt toxin into a field, you notice that the plants are becoming resistant to insects, but the spraying of the toxin made it less specific. So the idea was to introduce specifically the Bt gene into plant cell to engineer plants specifically producing the Bt toxin. Then, once the insect that you can see here is eating the transgenic plant, the toxin will be processed into the intestine of the insect. And there, it will be perceived specifically by some insect receptors that are present on the intestine surface of the insect. Okay? And this will create holes, pores in the intestine surface of the insect, and it will kill it. Okay, it will really affect the intestine of the insect and kill it. So because it's it's based on a specific recognition of the produce of the degradation of the toxin by a specific receptor that you can see here in red at the intestine surface. It's a system which is highly specific. It will only act on some insects, not all of them. So it's a kind of specific one, which makes the use of this Bt toxin useful to specifically target some kind of insect. And these Bt genes has been introduced in different crops, I told you, mainly corn and cotton. It was one of the first GMO plants to be commercialized, the Bt plants. They are expressing a gene allowing the production of a Bt toxin. When some specific insect eat those toxins, it's degraded, recognized by a receptor present at the intestine surface of the insect, and it will kill the plant. The second example, it's the example of the Roundup Ready plants. So what you can see in this picture, it's on this side plants that are cultivated with transgenic plants that carry gene, allowing them to be resistant to an herbicide, and they have been those plants treated with Roundup herbicide, and you see that only the crop, transgenic crop, here it's corn, will survive. And on this side, you see what will be the cornfield if you were not using an herbicide. So you see that the use of herbicide-resistant GMO allows to treat the culture and to keep alive only the corn. Here, all those plants that you don't want to grow together with your corn will use water, nutrients from the soil that will not be available for your culture, for your crop. So there is a use of creating those GMO resistant toward the specific herbicide. And here it's another example. It's it's the same example, but what this slide is showing, it's how proceeds herbicide to specifically keep alive the crop with each a GMO plant, GMO crop. So the glyphosate, it's what we call a broad spectrum herbicide, meaning that it will kill any kind of plant. Why? Because it's inhibiting one key enzyme present in all plants, which is the EPSP synthase. And this enzyme allows biosynthesis of amino acid. So if you inactivate this enzyme, plant will not be able to produce amino acid, then not be able to produce proteins, and it will die. Okay? There is a way to engineer plants that will survive the use of the herbicide, and that's what I will explain you now. to be able to produce aromatic amino acid, a plant is using one enzyme, which is, I told you, called the EPSP synthase. It allows the production of EPSP, which is a precursor for amino acid. When you use glyphosate, you inactivate specifically this enzyme, this plant enzyme, so the plant is not able to produce the precursor of the amino acid, and it will produce no amino acid and it will die. The plant will die. It's possible to produce GMO plant in which you will transfer an EPSP synthase, which is not of plant origin, but which is of bacterial origin. Okay, so the GMO plants that are resistant to the herbicide, they will of course express the plant EPSP, but they will synthase, but they will also express a bacterial EPSP synthase. Okay, the difference of this synthase is that it is not susceptible to the herbicide. So when you will treat the GMO plant with the herbicide, of course, you will inactivate the plant EPSP synthase, but you will not inactivate the bacterial EPSP synthase that you introduced into the transgenic plant. So this enzyme will act and allow the production of EPSP. The plant will use this EPSP as a precursor to produce aromatic amino acid, and the plant will be able to produce protein and to survive. Okay? So finally, you will be able to select plant by treating with the herbicide. All other plants that don't have the transgene will die. And that's what you could see, for example, here. Transgenic plant is carrying the bacterial EPSP synthase. It will survive. Other plants, they have only non-transgenic plants, they have only the plant EPSP synthase, which is targeted by the herbicide, and they will die. So this specific transgene allowing resistance to herbicide has been used a lot. If you heard about the Monsanto corn and so on, it's this kind of GMO plant. Okay. The next generation of agriculture transgenic plants on the market are more meant to improve the quality of the plant, and the most famous example is the golden rice, which has an increased content in vitamin A. I'm not going to speak about this today to save time, but when we will do the tutorial, we will analyze one article in which they describe the production of those GMO plants. Okay, so this will be for the tutorial. The transgenic plants that are more under development, it's transgenic plants that could be used to produce, for example, vaccines and all those planty cores to avoid the use of animals for making vaccines. A second kind of GMO plants that is under development, it's the use of transgenic plants to do phytoremediation. It means to clean some soil that have been contaminated, for example, with heavy metals. The idea is to use transgenic plants to trap pollutants from the soil, okay? To depollute some soil. So this is really more and more used also. And there are, of course, many possibilities of application to the use of GMOs. I would say a few words on the use of GMO. So it's more and more spread, the culture of GMO crops on Earth. I would say that there are the three kinds of countries depending on the use they make of GMO. In green, it's all the countries where GMO plants are grown in the field. And it's so there is, I would say, not much restriction to the use of GMO in those countries. And in those countries are grown those crops, cotton, which are BT cotton, resistant to insects, rice, resistant to pest, drought, or salt stress, maize, resistant to mainly to insects, and it's mainly BT maize, peas, peanuts, potato, tomato, and sugarcane. Those are the main GMO crops that are grown in those countries where it's allowed to grow GMO plants without too many restrictions. Okay? To give you an example, in India, which is the first producer of cotton, 96% of the cultivated cotton is GMO. Okay, so almost 100% of the cotton which is cultivated in India, it's GMO. And the same for example, soybean in America, South or Northern America. Most of the cultivated soybean now is GMO. So it's something I should be adding here. The same for crop. Most corn. Sorry, most of the corn cultivated in America is GMO now. Then you have the country in orange. It's the country like France. It's mainly Europe, where it's not allowed to grow GMO. It's not allowed to grow GMO in the field. There is one exception, is Spain. And in Spain are grown 90% of the GMO plants that are grown in Europe, because it's finally like the only country where GMO culture is allowed. And then you have the country in blue, including Vietnam, your country where the culture of GMO crops in the field, it's forbidden, but for research trials. So it's only in the blue countries, including Vietnam, only allowed to grow GMO in the field for research trials. Those crops that are cultivated in the field are not meant to be commercialized. So I don't know if it changed since 2016 in Vietnam, you know, or not? You have any idea or not whether it's allowed now to grow commercialized plants in the field in Vietnam? No idea?
I think
it's still
not approved.
Okay. Yeah, so
it's some research and also have some field to test in the real situation for the plant, but I
Okay.
don't think they have been approved in Vietnam.
Okay, so it's the same. Okay, so the same. Well, it's not the same in France. In France, we can grow GMO for research, but not in the field. For example, in my laboratory, I use everyday GMO plants for fundamental research. But we have some very specific laboratories with some specific security rules. Only people that have allowance to work with GMO can enter those laboratories. And those laboratories are really secured in the way that they are not opened to the outside. So, for example, no pollen, no seeds are normally able to escape from those laboratories. So it's secured laboratories that are isolated from the outside, not to spread GMO outside. Okay? So depending on the countries, you see that the rules are different. It's kind of evolving because laws may change. Because first you have a lot of economic pressure. So in Europe, for example, we are not allowed to grow GMO plants, but of course we import GMO crops, GMO food, sorry. We import a lot of products that are, in fact, GMO products. Not for human feeding, not so much. If you have GMO products, for example, I don't know, we may eat corn, GMO corn, but it has to be labeled. People need to know that they are exposed to a GMO product. And most of the GMO products, anyway, are not yet commercialized in Europe. Why do we choose not to grow GMO in the field? Because we don't know what may be the risk with GMO, and we think that there is a risk of dissemination of the genes into the environment through pollens, via insects, and so on. So that's why GMO plants are not cultivated in the field in Europe. And we don't know if it could have some toxical effect on non-target organisms. For example, we were speaking about the BT corn, the BT GMO plants that are produce those BT toxin. There is a lot of research going on the toxicity of those BT toxin toward not only insects, but also toward birds, other animals, or even human. And some research suggests it could have a toxic effect. Some research suggests it's not toxic, but as there is a risk, a lot of countries prefer not to grow those GMO plants. So it's kind of ethical, societal matter, and each country is choosing according to what people think is acceptable or not. For the moment, no change planned in Europe. The culture and the use of most GMO products is remaining forbidden. Okay, I skip this slide, and we will be moving to the next part of the lecture. I want to tell you because it's very important, very, very, very important. I want to tell you about the use of GMOs for functional genomics. I want to tell you how we can use GMO plants to better understand the function of genes. And what you must know is that we don't know the function of most plant genes, but maybe, and it's almost sure, there are a lot of genes available in nature that will be of great interest for humans, but we don't know their function. So there is a huge interest in functional genomics to better understand living plants and to better use living plants. There may be a lot of application, of course, for crop breeding, but also application in medicines and so on. Okay, so I will be speaking about the various application of agrobacterium mediated transformation in functional genomics. You need to understand the word genomic, and genomics, it's the scientific study of genomes. It's the study of the role, the function of genes. Okay, and knowing complete or partial DNA sequences and their function could be a useful information for us. And you must understand that the precise function of a lot of plant genes remain unknown. You must remember that a plant cell is carrying different genomes. There is the nuclear genome, which is carrying most of the genetic information. The nuclear genome is contained in chromosomes, okay, and it's carried, this nuclear genome, it carries most of the plant cell genomic information, okay? But then you have some additional genome in plants. Plasts have their own genome, and mitochondria have their own genome. But the function, a better understanding of the function of every gene is important for us. Okay? Remember that most of the genetic information is carried by the nucleus, but in a plant cell, you also have a chloroplastic genome and a mitochondrial genome. I told you a little bit about this before. A gene, it's made of DNA. We can identify different regions in DNA, a genomic sequence of one gene. There is a coding region with intron and exons. In most of the genes, you have exon and introns, and there are some regulatory regions that may be transcribed. There are some regulatory regions that will not be transcribed. It's the 3' UTR and the 5' region, which is upstream of the gene. In this region, you will find the promoter, which is involved in regulating where the gene will be expressed. this gene will be transcribed to give a RNA. Okay, DNA transcribed into RNA. You will have a premature RNA that will be spliced. And during splicing, the intron will be removed and will remain only the exon and the untranscribed region, the UTR. You will have what we call a messenger RNA. And this messenger RNA will be translated into proteins. Those are kind of basics on the gene structure that you must all remember. It's very, very important. It's kind of basic knowledge you must remember, you must know. Then you must know what is the genetic code. You remember that the open reading frame of the messenger RNA will be read by ribosome, and each codon in the RNA sequence will allow production of one specific amino acid. Okay, for example, UCU is encoding for serine. UUG is encoding for leucine. Remember this, huh? The messenger RNA will be read by ribosome, and it will allow translation of the nucleotide sequence into an amino acid sequence, and the amino acid sequence will constitute the protein. You must remember that the genetic code is redundant. So several codons may encode for the same amino acid. Okay, for example, you see here, you have a list of 1, 2, 3, 4 codons that are all encoding for proline. So the genetic code is redundant. Then you have codons that will allow initiation of the translation. It's, for example, this codon encoding for methionine, and you have termination codon, stop codon, that will stop the translation. And you have 3 different stop codons, here, here, and here. This will allow the translation of the messenger RNA into a protein. A lot of plant genomes have now been fully sequenced, but it's something which is really recent in the history. The first plant genome to be fully sequenced was the genome of Arabidopsis thaliana. The genome of Arabidopsis thaliana was made available in 2000, so only 20 years ago. It's a rather small genome, you see, if you compare to the human genome, which was fully sequenced, the first version of the human genome, about the same time, in the 2000, more or less. You see that the human genome is much bigger than the Arabidopsis thaliana genome. However, on the human genome, you have a bit more than 20,000 genes. On the Arabidopsis thaliana genome, which is much smaller, you have more genes, 25,000 genes. During the last 2 decades, a lot of plant genomes have been fully sequenced, and the most important crops have had their genome sequence. Rice, poplar, maize, soybean, potato, tomato have had their genome sequence. More recently, I think it was 2018, and a more recent genome has been made available. The genome of wheat has also been made available, which is one of the most complex genomes. I want you to check those numbers. You see that the size of the plant genome is not really important when you compare to the plant genome. However, plants encode for much more genes than the human genome. Why? One reason may be that humans, animals, we are able to move. And if we are exposed to a stress, we may be able to run away from this stress. At the opposite, plants are not able to move, and they are exposed to changing environments. Environmental conditions are changing always for plants. The weather may change. Plants may be exposed to drought, to frost, to cold, and they have to be able to adapt very quickly to those stress. Plants are exposed to many kinds of pathogens, and they cannot run away. So they have to resist to those biotic stress. And one reason why they have so many genes may be because they have to quickly adapt to changing environments. That we don't need to do. If we are exposed to a stress, we may only have to run away. Plants have to adapt to their environment, and probably the huge diversity of genes they carry helps them to adapt to their environment. That may be one reason for this huge number of genes. Remember this, a genome size is not linked to the number of genes, and some small genomes may encode for a huge amount of genes. Okay? Plants have small genomes, but those genomes encode for a huge amount of proteins. Okay? Similarly, the DNA quantity is not related to the organism complexity. This is also something important to remember. You have some very simple organisms that have very complex genomes. The use of genomics can help us to understand how genes affect one another in different species. So it's very important to understand genome structure. To identify one gene involved in a disease process or an abiotic stress may help us to develop resistance toward those stress. Similarly, it's very important to understand the function of genes that may be involved in cell wall biosynthesis, plant oil biosynthesis, and so on, because it can help us to improve crop lignin content. It's very important for paper factory. Oil content, I told you, this may be very important for the production of bioesters and so on. The better we know the function of plant genes, the better we may be able to use available plant biodiversity. I want to say a few words on model plants. I told you, most probably Arabidopsis thaliana is for sure the most used plant to do functional genomics. Why? I told you because it has a small genome. The genome is fully sequenced. The short life cycle is really short, 8 weeks maximum. It can be shortened, and it produces a lot of seeds. A lot of mutants are available, so we can easily study the function of a gene, and it's very easy to transform. Okay, but don't forget that more and more plant genomes have been sequenced, and more plants are becoming plant models. I told you it is very important to understand plant genome structure. There is one notion I want to introduce. It's the notion of synteny. Synteny, it's kind of typical for plants. Okay, plant genes are organized on chromosomes in a certain way. And very often, inside the plant family, the order of the gene in a chromosome is conserved. So the more deeply you know the structure of a plant genome, the more you can predict what will be the gene function of a relative to the plant species you have been describing. Okay? Because the structure of the gene is a kind of conserved inside plant families. That's what we call the synteny. This is one important slide. It's something I was speaking about previously, is that we know the function a lot of plants gene, and if you consider Arabidopsis thaliana, you see that still there is a lot of gene for which we don't know the function. I told you Arabidopsis thaliana, it's 25,000 genes, but some crops, it's more than 40,000 genes, and the proportion of genes for which we have no idea about the function is even bigger. So there is a need really to better describe the function of plant genes. And that's why functional genomic tools are really required. What I propose before to describe how we can use functional genomics to better identify plant gene function, it's to make a last break. It's for you, it's probably 38 past 4. It's probably 4:39 for you. We will make a break until 10-minute break, okay?